Teclistamab is a bispecific antibody. So it's an antibody that has two specificities. So one specificity is for BCMA, or B cell maturation antigen, which is a cell surface target on multiple myeloma plasma cells, against which a huge range of different therapies are in various stages of preclinical development. And of course, we recently had approval by the FDA of the first BCMA targeted therapy to make it to FDA approval. So one specificity is for BCMA, and one specificity is for CD3, which is a molecule in the cell surface of T cells. And so the way teclistamab works is it binds both the T cells and to multiple myeloma plasma cells and brings them together in a way that forces the T cells to recognize the multiple myeloma cell, and the T cells are activated in the process, and so you get killing of the multiple myeloma cells by the T cells that the antibody engages. So that's the proposed mechanism of action. So the patients were multiple myeloma patients who were relapsed refractory. So they had been exposed to multiple lines of prior myeloma therapy. So the patients on the study had a median prior line, median of 6 prior lines of therapy overall. And most of the patients on the study were shown to be refractory to the major drug classes that we use to treat myeloma, so-called triple class refractory patients, meaning they had progressed and shown refractoriness to proteasome inhibitors, thalidomide analogs like lenalidomide and pomalidomide, and an anti-CD38 antibody. So these patients had been exposed to most of the active drugs that are used to treat multiple myeloma, and their disease had been shown to be refractory to these drug classes. And so that's sort of a typical phase 1 population for multiple myeloma, but a patient population that was pretty heavily pretreated. It was a fairly well-tolerated drug. The most concerning side effect that we saw was cytokine release syndrome. So this is an inflammatory reaction that is due to the activation of the immune system against the myeloma cells. It's similar to the cytokine release syndrome that's seen with CAR T cells. To try to mitigate this, patients received, before they received the first full dose of teclistamab, they received a couple between 1 and 3 smaller doses of the drug in the week prior to the first full dose, to try and ease in the intensity of that reaction. And so we think that mitigated the risk of high-grade CRS, and most or all the CRS that we saw was grade 1 or 2. And in that respect, it was very manageable. Patients were hospitalized for their first few doses to monitor for cytokine release syndrome, and many patients required some treatment to mitigate cytokine release syndrome, either supportive care or in some cases using the anti-IL-6 receptor antibody tocilizumab, which reduces that inflammatory reaction. So that was, I think, the most significant adverse event, and was seen quite commonly, in 55% of patients, including 64% of patients who were treated at what we determined to be the recommended phase 2 dose of the drug. We also saw occasional cases of neurologic toxicity. So that's another toxicity that has been seen with therapies like CAR T cells and bispecific antibodies that induce a large-scale activation of T cells. There were 2 neurologic toxicity events that were grade 3 or higher among the 149 patients that we've studied so far. However, none of the patients in the more recent dosing cohorts that received subcutaneous, no high-grade events among the patients who received subcutaneous dosing. One very low-grade event, incidence of tremor. And so these neurologic toxicities were reversible in all cases. And so that remains a toxicity that we have to keep an eye out for, but fortunately it was very rare, just 5% of patients, mostly low-grade events, although a couple occasional events that were grade 3 or 4. It's very encouraging so far. So, this was a phase 1 study, and so patients were treated at a wide range of doses, ranging from very low doses up to what we eventually determined was the recommended phase 2 dose. And so, still learning about how people do over the long term. And in a phase 2 study with a much larger number of patients who are all treated with the same dose and schedule will ultimately give us a better estimate of how likely patients are to respond. But what we've seen so far is very encouraging. So patients who were treated at the more active dose levels between the intravenous and subcutaneous cohorts, across those higher dose levels, the sub-Q and IV dosing had a response rate of around 70%. The 22 patients who are evaluable at the recommended phase 2 dose had a response rate of 73%. So that's a very high response rate for an agent in this type of patient population who had received so much prior therapy, relapsed refractory myeloma. And so, yeah, it does seem to be working well, although ultimately that requires confirmation in larger studies.